ulcerative dermal necrosis, UDN. Now this is not the name of any bacterium or virus, it's simply a description of a set of symptoms. Ulcerative simply means with ulcers, dermal skin, and necrosis is death of part of a living body. And you can see that on this fish caught last year in spring. On the head, part of the body where there are no scales, just the skin, you can see a white area of discoloration, and in the middle a red patch where an ulcer or lesion has formed and is eating through the skin. Now this sort of thing matches a description from the 1970s, and you can see on this other fish caught this year patches of grey or whitish discoloration on the skin, and just in the middle of the largest one the beginnings of the red ulcer eating its way through the skin. This is a fish that was fresh in from the sea and actually still had sea lice, and this is the point about UDN style symptoms. They are on fish coming in from the sea already with the marks on them. And it does appear that what is being seen now is similar to what was seen in the 1960s. Those blotches, discolorations on parts of the body that don't have any scales, and then the ulcers beginning to form in those, and of course coming in the early part of the year when river temperatures are low. Now in the 1960s it was suspected there had been a previous outbreak of what they were seeing back in the 1870s, and that had been investigated by a parliamentary inquiry. Unfortunately, with the state of knowledge of fish diseases at that time, there really wasn't very much they were able to say about it. It's interesting though that it did seem to be confined to the Tweed, the Eden and the other Solway rivers. What caused a great deal of confusion then was the appearance of fungus on these sick fish. Nobody could agree on whether the fungus was a primary cause of the problem or whether it was in fact a secondary infection that had been allowed to colonize the fish through some other infection. Now, fish with these sorts of symptoms are always present at a low level, but this report from 1905 is of particular interest as it appears to describe the very first stages of UDN while the fish are still in the sea, long before the red ulcerations begin to appear. And they were calling this a white spot. Now, it's interesting that they say that the symptoms that they're describing would have been more widespread had there been more sunshine. They seem to associate the appearance of these fish with these symptoms with bright sunny weather. And that's particularly interesting because one of the theories what UDN is that it is a sort of sunburn. So what is UDN? Is it an infectious disease or a skin condition, like eczema in humans is a condition of the skin, not a disease? If it is an infectious disease, it should obviously be possible to infect healthy fish with material from sick fish and then see the same symptoms appear as a consequence. But this was not successfully done in the 1960s as reported in this paper below. In fact, having failed to transfer the disease from sick to healthy fish, they actually sent material over to America where there were better facilities for them to try to do this. But again, they actually failed to infect healthy fish using material from sick fish. And a Danish laboratory was also asked to try and also failed. Now, some new information or a new view on UDN that wasn't available in the 1960s is now available from the experience of salmon farms. Because if UDN was an infectious disease of the sea, then surely it should turn up in the cramped and crowded conditions of salmon farms like so many other diseases have. But actually it never has. This is a suggestion, a piece of evidence that UDN is not an infectious disease. But on the other hand, the outbreak of UDN in the 1960s seemed to follow a pattern, and that looked like a spread of an epidemic, because it began in one area, the southwest of Ireland, and then spread to others, moving northwards and eastwards, and finally reaching the Tweed in 1967. So there's evidence of sorts on both sides of the question of whether a UDN is an infectious disease or not, and the plain answer is, at the moment, we do not know what it is. Once flesh is exposed by the ulcerations, it can be colonised by spores of the white Sapolygia fungus, which can then get a grip and grow and make the white patches that make these fish so visible. If the fungus gets a real grip and destroys more of the fish's skin, the fish can lose the ability to regulate the amount of water entering its body, and that can kill it. And it's this fungal attack that results in the dead fish that are seen early in the year when there is an outbreak of UDN. However, it is very important to be quite clear that the white fungus is not UDN. There's a lot of confusion over this. Many people think it is. UDN is the ulcerations that let the fungus into the fish. Fungus is therefore a secondary infection, and it is that can have the fatal consequences. However, as water warms up and the fish itself warms up, it can actually fight off these fungal infections and indeed the underlying ulcerations and recover with only scars to show for its experience. It's interesting to look back to reports made in the RTC's annual reports in the 1960s and 70s. In 1967, for instance, the first signs of UDN were recorded, and they were in fact on 2 fish caught in the nets on the 14th of March 1967. From that date, it says, the disease increased in severity until a flood in late May, after which it virtually disappeared, until it reappeared, they think, again in November and December in the shape of very high mortalities of fish. Now, not only were these thought to be greater than the usual post-spawning mortality, but there were many unspawned fish amongst them as well. However, the report also says that spawning in that year, 1967, was, as far as they could see, as good as in any normal year. Some of the spawning fish showed slight signs of the disease, but spawned normally. In the 1972 annual report, it says that the incidence of UDN appear to be at about the same level as that of the past few years. They also point out that the very large numbers of salmon that were entered in the river during the year were actually nearly all the young of fish which had spawned during the outbreak of UDN. So they weren't actually seeing any signs that this outbreak was actually affecting stocks. So what effect did it have on stocks and catches in the 1960s and 1970s? One way we can look at the possible effects of UDN in fish stocks is to look at the catches made by the nets during this period. Here are the February to June catches of netting stations in the lower Tweed from 1950 to 1979. These are not nets in the estuary, these are nets in the actual lower river itself. And if you look at the 1967 catches, spring catches, you can see they're not actually much different either before or afterwards. So there is no actual sign of any effect of UDN on the numbers of fish coming in spring. Now, there was a big drop in spring catches from the late 50s into the early 1960s, but as you can see, this happened a few years before the first appearance of UDN, so presumably is not related. And if we look at the annual catches of these lower river nets, not just the spring catches, you can see that there's no sign at all of any impact on stocks. In fact, there's a general upward trend over this period. Now, if we look at the actual salmon rod catches of 1950 to 1979, you can see that in 1967 there was a very big drop in catches, although this is exaggerated by the fact that the previous 5 years had had exceptionally high catches. In fact, the total annual catches in the late 1960s were not much different from those in the late 1950s, before these very large catches were made. One thing that does change, though, is the balance of the catches. Now, the columns in black are the numbers caught before the 1st of July in the first half of the season, and in grey are the numbers caught after the 30th of June, the second half of the season. And you can see that in the late 50s, far and away more fish were caught in the first half of the season, the second, and this changes in the late 60s. And this transition is happening in the early 1960s, when the catches become 50-50 between spring and autumn. And this, of course, is the time of the very big and very noticeable switch from most salmon coming back in the spring, in the first half of the season, to most of them coming back in the second half of the season. Now, it's sometimes thought that this changeover between the seasons was attributable to the effects of UDN, but as you can see from this changeover was happening in the early 60s, before the first appearance of in 1967, so presumably is not connected. So that drop in spring catches from 1967 onwards may be something to do with UDN, but it may also simply be the system has switched from being mainly spring stocks to being mainly autumn stocks. Now, as a result of the numbers of fish with symptoms what appear to be the UDN of the 1960s that were seen last spring, the River Tweed Commissioners, the Spey, Dee and Nith Fishery Boards, and the Association of Scottish Fishery Boards and the Scottish Government have joined together to support a PhD student at Stirling University to look at these fish with modern techniques. However, there have been far fewer fish seen this year with the symptoms, so only 4 fish have been sampled so far. The sort of fish that are needed for sampling are really quite specific. You really want them to be freshly killed, ideally immediately before the samples are taken. You want them to be fresh from the sea, so they're not contaminated by freshwater organisms, and with no fungus preferably, or just a very little, so that anything found can be attributed to the UDN and not to the fungus. Now, the first samples taken under the protocol are from the skin. This is done by taking swabs. Swabs are little cotton wool buds, rubbing them against healthy skin and also the ulcers, the lesions, and then putting that material onto these agar growth plates. Now, agar is a sort of jelly to which different chemical nutrients can be added, and depending on which nutrients are present in which plates, different sorts of bacteria will grow if they were present in the swabs, and if the bacterial colonies grow, they can then be identified. Samples are also taken from inside the fish, from the liver, the heart, the spleen, the kidney, the gut and the stomach, and this is for testing for viruses, for examination of cell structures. So the hope is that with modern techniques and these samples that we're taking, we will at long last be able to find out what exactly this thing called UDN actually is.